What's up, guys? So, it's been a minute since we've been on, but really excited. Got some amazing updates, some really cool technology. I'm so excited about the stuff that I've learned in this last month. And it's just ironically crazy, but a lot of the stuff that I learned has been from my students. It's a trip to sit here and think about. I'm the teacher, I'm the one educating them, but at the same time, a lot of my students have been coming through and bringing literature and just they've been studying, so they have different knowledge, different aspects, different outlooks, different takes. And it's really cool that, like I said, I've learned so much. And the point of that is a lot of people that came to class in the beginning, unfortunately, didn't get to learn a lot of the stuff that's taken me quite some time to learn. So the beautiful thing is anybody who has taken the class can always get back to me and I'll gladly share or schedule a day you can come back for a refresher. And I'm happy to share all the latest stuff that I've learned because it really is awesome. I've learned a lot of stuff and it's worth it to give me a call if you want to brush up, kind of touch base, a lot of really interesting stuff. I want to say thank you to my students. I've had like 6 students over the last week and a half, 2 weeks. We were kind of behind and kind of started cramming people in. We had a fire at our lab a year ago. It's taken some time for construction, so we've had some logistic issues, but we're finally getting students back through. And I wanted to say thank you to Vinny. Awesome. Did a great job. It was so much fun talking to you about the applications you're trying to administer and the things you're going to be doing. Super exciting. And then Marco and Gabe, just awesome. All my students have been coming through and inspiring me. So Vinny, Marco, Gabe, Tim, Mike, some of the most recent students, and I feel they're very fortunate because they've got to learn some of the most recent technology that I've learned after basically 6 years of studying now. It's funny how a lot of the stuff we go through failure, it sucks during the time of going through. I look back at how I was so close so many times to quitting in tissue culture because I was failing. I went to class. I thought I was just dumb, didn't know what I was doing. But long story short is it sucks to go through so long with something and then to have it fail. It just inspired me to be a teacher. Literally, we shouldn't have to go through something that long, but regardless, you have to put the hours in. One thing I've learned is failing has been a huge lesson, a valuable lesson. Without that failing, I wouldn't be able to create protocols now the way I do, understanding cytokines and auxins through the failure, through the time spent making media, making mistakes. And I kind of wanted to touch on the whole tissue culture kits because so many of my students have been coming through and they're using kits and they're failing or having trouble. And this isn't to knock the tissue culture kits by any means because I started with one. The whole point of this is to teach you that you really have to understand the chemistry. You really want to understand the chemistry before you get started in tissue culture. Understanding cytokines and auxins, the hormones, are critical. When you're buying a kit, you have to understand you're using basically a one-size-fits-all, and that does not work in tissue culture. I'll be the first to tell you, you have to have an understanding of chemistry and how to make small micro adjustments to each strain. Yes, you can get them in with a one-size-fits-all, but usually they're ugly. You get epigenetics, things don't look right, and it's hard to be consistent. So until you understand cytokines, auxins, how they interact with each other, what each one does and how they react to each strain, save your money. You're gonna put all your strains into these kits, and I'm seeing it from my students themselves. They've lost tons and tons, thousands and thousands of dollars in their genetics because they put all their stuff in, not knowing, they're thinking it was going to work. And I'm sure everybody has good intentions, but this whole video is about understanding, the things you need to know before you get into TC and waste your time. And again, it's not a waste of time failing, but a lot of people park the kits and they never go back. They spend all this time, and after 6 months, if you're not doing something, making money, being efficient, who can do that? You're gonna go back to what you were doing or something else, because most people will not put up with failure past a 6-month period. They're gonna be out doing something else. And the secret to tissue culture is it takes minimum of 3 months up to 24 months per strain to get them through stage 1. Stage 1. That's a long time. Most people have no idea how long it takes to chase, clean, and eliminate bacteria from inside of the plant, which is what the reason most plants deteriorate, lose vigor, lose terps over time. It's because they're full of bacteria. So how do you get rid of bacteria? Obviously, sterilizing the plants before they go into TC is huge, but also the inputs you give your mother plants are huge. The things you're feeding your mother plants can come out in a bacterial form, endophytes in culture. So be mindful of what you're feeding your plants. But really, it's about understanding how to subculture. No one really talks about this in the kits, and you don't hear it in the Bill Graham stuff, but subculturing is simply changing the media on a regular interval. Whether you're getting infections or not, sometimes if you're getting infections, obviously faster, but if you're not getting infections, it's about a 12 to 14-day interval, meaning you're making a new medium, taking that plant and swapping it out, cutting the base off, cleaning it up, and starting the process over. Some strains can get through with 3 to 6 subcultures and move on to stage 2. Some can take 10, 15. I mean, the numbers can be outrageous for how bad these plants can be contaminated, and it just takes time, and there's no rush, there's no race. This isn't a let's get plants into culture as fast as we can and out because the boss needs me to put, clean plants in the garden. Tissue culture takes time, and no matter what your situation in your garden is, you really have to put a lot of time in first to build the lab, 6 months to a year. But once the lab is up and running, it's an investment that no one pays attention to because no one's willing to risk the amount of failure to get to this point, which is about a 2-year period. If you can withstand going through the failures and creating protocols and making yourself just enough successful, not racing to get to stage 2, stage 3, and get plants into the garden, but into stage 1 until the plant's fully clean, no infection, multiple sources of indexing. Index meaning we're doing full pathology, microbiology, everything we can possibly do to that plant to ensure it's clean in stage 1. Does not go past stage 1 until we have passing grades on all the pathology. Most people don't even do that. They're in such a hurry to get to stage 1 to stage 2 to root, to get it out, to say, see, look, I did tissue culture. That's not tissue culture. It is a slow withdrawal of internal contaminants that takes time, and each strain is variable on the contamination and how long it takes to acclimate to culture. This is why understanding cytokines and auxins, because it's so important. While a kit might get you a few months in before the plant finally wilts and dies, understanding cytokines and auxins will allow you to make micro adjustments by looking at the plant and creating ideal solutions that make the plants thrive and survive. It takes a lot of time. And again, most people don't want to go through that failure, or it's hard to sit there. I mean, I don't know how I did it, honestly. I'm surprised that I did. Thankful that I did now. But the point with TC is you're going to have to spend some time. You can't buy this. You can't have it given to you. You literally have to spend the time failing without going broke. Don't go out and buy a kit until you've studied cytokines and auxins and you understand endophytes and internal bacteria. Then go buy good equipment, decent equipment, instead of buying this kit, which is a hobby kit. I'll tell you right now, I totally respect people going out and doing it and getting fired up to do it, as they want to do TC, but this is not a hobby. If you're going to treat it like a hobby, you're going to get hobby results. If you're looking to do something to really increase your business, your potential, cut costs, limit all kinds of problems that happen in normal grows, and actually be profitable through indexing and data collection through clean plants, do it the right way. Invest your money into a good hood, good autoclave, a good microscope. You need these things, a meristem, to perform normal tissue culture. And again, it's not a race. It's not how fast you can get it in and get it out. 3 months to 24 months is an average time to get through stage 1, just to get through the infectious part. And that doesn't include the testing, the pathology, indexing to make sure that everything's been cleared before we multiply that plant. We don't multiply a plant until we know it's got certification. It's been cleaned, it's been pathology, chemistry tested, and then electromagnetic magnification with reactive agents that glow certain colors based on whatever you're looking for. You really want to have some source of guarantee before you multiply the plants. And I don't know many people that do that. There's a few people who are professional and do it. We're bringing all that stuff in-house. We have a class on Tuesday up with Kind Brothers and a few of our students. We have My Flora DNA coming in, and they're going to be giving us all the equipment and showing us how to do basic hops testing along with more detailed data. There's two sources of gathering this, a yes and no, and then there's like getting really critical data, basically how much of a load. So we're going to be learning both of those, and that's exciting. And we're also going to be utilizing Toomey, their testing software in their platform as well. So it's really going to allow us to track and keep data and really start measuring not only genetics, but how effective our treatment, just some really cool stuff. So back to the creation of protocols, understanding your cytokines and auxins. Can't emphasize enough. When I say cytokines and auxins, I'm talking about like TDZ, benzamidopurine, kinetin, 2IP, metatopoline, zeton. Those are things that make the plant grow. And then auxins, IBA, NAA, IAA, 2,4-D, just to name a few. Those are really your basic auxins, but it's the mixing of the auxins and the cytokines and the ratios in which you do that, understanding them is critical. And that, again, can't be given or be bought. You literally have to spend time studying what each hormone does to each plant, each food. It's a process. It takes time. So again, the kit is something people are kind of rushing into, thinking they're going to save their strains and not even understanding how to subculture into future mediums and how to index before you go to stage 2. If you're going to do this, we just want to see you do it right. There's a reason we're expensive, and it's not just to be expensive. We're expensive because we believe in the service we offer will make you successful. We're not here to sell you a dream or a kit that's going to sit there and get parked. I want to actually sell you the knowledge to be able to create through identifying over time, being able to look at the plants and make your own solutions to make and master protocol creation. When you're able to do that, now you can literally pick every detail of the plant, control it through hormones and auxins. And really, it's like I said, it's unlimited when you get to that point. But you have to understand these infections can take 2 months, 3 months, up to 6 months, sometimes a year. And when people buy these kits, they don't understand that they see all these infections and they do not know how to get out of it. And they just keep killing and reinfecting over and over. So people that have been buying kits, I've seen so many of them coming through with the students. You can call us. I'm happy to give you some at least a little bit of understanding. I've done this for a few of my students. I've given them media solutions and made some protocols for them. Just enough for them to go test it, have a little bit of faith in us. At that point, if they feel like calling us or coming to class, it's a little bit easier to trust once we give you a little bit of success. And that's all we care about. We want to see people successful. We're in this to make people successful. Your success is our success. So I'm interested in teaching you creation of protocols, understanding cytokines and auxins, and indexing, meaning we do all this work to stage 1. After that, now we do all the pathology and just to make sure we don't replicate any kind of internal pathogens, bacteria, viruses. That's the whole purpose of tissue culture, to make sure we're not multiplying sickness, bacteria, or virus. So most people that are doing kits, don't get me wrong, I totally respect and appreciate it again, but they're not doing these things. It's a learning experience. It's a hobby. Totally get it. But for the people who are serious, need to make a lot of clones at volume for their business, for their legal operation, whatever it may be, just to preserve strains. That's what we're here for. I want to show you how to be successful, cut out literally years and years of failure in pulling out your hair and give you certainty in tissue culture. I believe in myself. I believe in the company. If you're having issues, if you bought a kit and you need some help, leave a comment. I'll answer it. I'll help you out. I'd like to steer you in the right direction. Understanding protocols to get you going in the right direction, because at least if you're beginning to go in the right direction, you'll get an understanding of what you're doing. You'll be able to start spotting things and noticing things, and over time you'll be able to create your own protocols. And at that point, it makes it a lot more fun instead of pulling your hair out and not knowing. So Bacteria and infections are going to happen. Unless you know that going into buying a kit, you're not going to know what to do. And it's literally understanding how to subculture, meaning don't go a month or two before you change the medium out. Every 12 to 14 days, pull that medium, make a new medium, take that thing out, even if it's not sick or anything, recut it and stick it back in, clean it up. Repeat that process over and over. That's what tissue culture is. The more often you cut and replace that plant into a new sterile medium and start over, you're essentially regrowing new material, outrunning the old sick, bodily, infectious part, recutting it, replanting it. This is why clones work to a degree without TC, because you're cutting the tips. Minimal infection lies towards the meristem. About a half an inch from the meristem is where viruses lie. So a half inch below the meristem is where the virus lies throughout the body. So if you can take the end little half-inch tips, you tend to be free of virus or pathogen. So it makes perfect sense. But even more so, if you take small pieces, wash them in bleach, and then regrow them over and over in a sterile medium, you can outrun a virus. But it takes a lot of time, a lot of subculturing, understanding the food. They go through a lot of morphological and physiological changes during this time. Requires food and hormone adjustment. Otherwise, you'll get weird, funky growth, yellow, funky shit. Understanding how to create little keys with cytokines and auxins at different ratios, understanding the measurements, the pipette, micromole measurements are critical. It took me forever. I wasn't good at math when I started all this. Wasn't a high school graduate, didn't go to college. So understanding a lot of the chemistry, a lot of the measurements was difficult for me. We have a lot of links, a lot of new information now. We can get that stuff to you and really help you past a lot of the sticking points in infection, hormone dosing, and measurement, so you can do your own. Don't buy media from other people. Don't sit here and guess. You literally have to understand why you're doing this. If you don't have an interest, don't do it, because you'll just, it's spinning wheels. You have to really understand why each hormone does something to the plant. You have to have that desire to understand. If you do and you follow it through, it opens up other doors that you never thought possible in chemistry. In growing it, to me, it transcends cannabis, because you can pretty much grow anything at that point and create your own protocols and solutions and store everything on a bread rack efficiently and have it really out and growing in a matter of 15 or 20 days, free of virus, bacteria. And you're talking about fun to grow again when you grow a plant that doesn't have any problems. This is why you've heard the term seed rate growth. That term comes from a plant that doesn't have any internal bacteria, pathogens, and virus. They tend to thrive. That's why seeds do well. Here's the problem. A lot of the seeds that were bred over the last 5 to 8 years are sick, straight up. We're testing seeds, and a lot of the seeds that are going through are coming up sick. So don't think you can just pop your way out of this. It's coming to a point that if you're not doing some type of meristem, if you're not doing early DNA testing on seedlings and ensuring that you're not introducing those to the garden and reinfecting and vectoring, everything has to be grown in a test tube, tested early on, indexed, verified, then it gets multiplied. If you're not following those steps, it's a sure way to eventually end up with a problem in your garden that you're going to be faced with failure again. So take the necessary steps, do it right. The kit's cool, but you really want to focus on being successful. This is about a long-term commitment. If you're willing to do that, I assure you, it's well worth the commitment to invest the time and go through that little bit of failure, because it's really not failure. That is the learning curve it requires to understand chemistry in the food making of this whole journey of tissue culture. Until you get that, it's taken me a long time. 6 years of hardcore teaching, study, to finally kind of grasp the process. I want to share that with people who are going out there and spending all this money on these kits. It's frustrating, because I just don't like seeing people go through the same thing I went through. I never intended to be a teacher. I became a teacher because of the sheer frustration of the information that was and wasn't out there. We just want to steer you in the right direction. Be sure to leave a comment. If you have anything, leave it in the comment. Follow us, subscribe. I'm going to be dropping a lot of knowledge as much as possible, and I'm excited to share with you. So again, go learn about cytokines and auxins as much as possible. Study what they do, what they're made out of. Study the ones that are organic and synthetic. Learn those, and then study internal bacteria infection in plants, endophytes. No one ever talks about that stuff. It's rare that you hear about it until you buy a kit, until you see all this stuff happen, and then you go, shit, I better figure this out. Then you're scrambling. Study beforehand and gain some knowledge because you can make the kits work. You really can, but you just got to have a really good understanding on what to do. You're going to have to buy some other things. You're going to have to be able to adjust and create different pH, different types, different media. There's lots of variation in tissue culture. It's crazy how much variation from pH, from sugar to food, to hormones, to mixing of hormones, to temperature. You're talking about so variable that's very hard to really hone in and create, perfection, very epigenetic free growing. It makes you think you suck at growing because the plants look like hell. So pay attention to bacterial infection and endophytes, and pay attention to cytokines and auxins. If you're going to go buy a kit, do your homework first, understand these things. At least you'll get a little bit successful and you won't park the kit. If you're thinking about buying a kit or you're thinking about doing TC, give us a call. We're happy to talk to you and give you all the lowdown, the benefits, the good and the bad. We'll be straight with it, because look, if you're not willing to invest time and the money in yourself, we don't want you as a student. No problem. But if you want to move along fast and pick this up, figure it out, make your business successful, I fully believe this. This isn't a sales pitch. I've been doing this since I was 14. I'm 52. If you're not doing TC or you're not doing the pathology in-house, someone's going to be doing it for you. And you're talking about a lot of money to do all these tests, especially the small stage, through middle, through end stages. You have to be tested all the time now. You'll go test broke. So learn to do it yourself, learn tissue culture, learn this process. Otherwise, again, with AI technology now, believe it or not, they have machines making media without human hands. They're like almost like a dosatron connected to all these hormones and media types. And AI comes in now and creates protocols based upon past, present, and future success. You have to understand that unless you're on board and getting this stuff, learning, you're going to get left behind. Again, not a sales pitch. I've been doing this for long enough to know I've been able to adjust the curve of learning through cannabis, from clones to TC to now pathology. Understanding the pathology is the future of growing. Again, it's a lot of money to do tests on plants, but in order to grow successfully and eliminate all the problems we're having, you're going to have to understand pathology. And that starts with tissue culture, keeping your plants isolated, free from bugs, viruses, and vectors. The ability to test small, full, whole plants. Again, once you do this, I can't tell you enough, it's fun to garden again when you go through all the stages of tissue culture and you come out with a plant that's clean, tested, verified. It makes you feel like you're really a super good grower. And that sounds cheesy, but it really makes the plants thrive. And it's crazy because you get yields, you get terps. We're all about having success and being productive and successful. I'll tell you right now, this has made such a difference. It really makes me want to grow again, but I don't. I'm trying to get out of this business eventually, do something to retire and maybe focus on protocol creation. But for people who are trying to make this into a successful business and stop loss and create, specific libraries and strains that are tested, I'm telling you right now, the shit I've learned in the last month is crazy with artificial intelligence, creating mediums without people. They're going to be making solutions so precise and pinpoint that literally it's going to change the game. And there's companies like Conception and Nodes, other ones that are out there, they're doing it. They're actually in this process, and now it's data collection. They're collecting data, and the data is going to be used to completely revolutionize the industry through artificial intelligence. So it's coming. If you want to get on board and learn this stuff, it takes 6 months to a year to get this going. So give us a call. If you're trying to get in and get this going, you can come to class, 2-day class, or we'll come to you. If you have a lab, even better. We'll help you build the lab. We'll come teach you in your lab and show you what to do to get you on the right track from the get-go. Cut out a year, 2 years, and then we can also get you started with clean genetics. So people considering buying TC kits, awesome. Just be aware. It's a lot of money. For the amount of money you'll spend on that kit, you could buy a good hood, a good autoclave, and a good microscope, and actually have a nice lab. You need a good autoclave, you need a good hood, you need a good microscope. So when you have questions, leave the comment, call us, hit my website, Epigenetics.com. We want to help. We want to help people succeed. That's what we're here for. So study cytokines and auxins. Really get into the endophytes and the bacteria. If you want to have success in tissue culture, Don't just go into it hoping, you know, someone's going to give you the answer. There's no magic sauce in tissue culture. It is literally you understanding, like a NASCAR, that's the Okie in me, understanding that it requires a set point, kind of a flat baseline, and then you go around the track and you make micro adjustments to get that perfect medium. It takes a lot of time, a lot of effort, but the reward in doing so is a plant that's ultra healthy, ultra clean, and grows like you've never seen. Literally, that just happened around, but really they grow like I've never seen. The terps on these plants, I only say it through experience now. I wasn't a complete believer when I started. After I run these plants now and see what they do after tissue culture, like I said, it's fun again because they yield, the terps are killer, and these are the plants we want to breed with. We want to take plants now that we put through the entire process of tissue culture, merification, we're going to call it, the process we're developing, which is meristem process, followed by pathology, PCR, chemical testing, multiple sources for quantitative and just reactive, yes or no, followed up with reactive dyes and electromagnetic microscope so we can identify where. It's like identifying you have cancer first, but then identifying where it's at so we know where to work on it. And then we can say, okay, yes, we can fix it by cutting the head off and regrowing it. And then after, we can come back and see, is it still here? Did it grow back? Did we fix it? So now we will have merification, which is a meristem process with multiple sources of verification of being clean and free of virus and pathogens. If you're not doing that, good luck surviving in the industry. Doesn't mean you can't still do stuff. Great. But I'm just saying in the direction it's going, for me being in this as long as I've had, I'm telling you right now, things are about to change, and they already are. If you want to get on board, call us. We're glad to get you a head start. We're excited to share with people who are interested in building their own laboratory and building their own personal knowledge, and I'm excited to share it with you. Again, thank you for listening. I appreciate it. I'm going to be doing another video in the next day or two, more about the hormones, in depth about the meristem, and really kind of breaking down the AI technology, because like I said, it's really crazy. Here, I thought I had to sit here and create these protocols and solutions, which we do, but it's coming to a point where now there'll be software in a machine that can literally make some perfect stuff. So I have a feeling that the industry is going to rapidly see some changes. Better to be on board now than sit around on the fence. Do something. Make yourself smarter, make yourself better. Even though times are tough right now, it's the people who invest in themselves and are prepared. The market ain't going away. People still consuming and into cannabis. There's just a lot of people who quit. There's a void. Get smart, educate yourself, fill those voids, and make yourself a better business, better success by eliminating all the problems that are plaguing everybody else. Thanks for listening. Hit the subscribe button, follow us, leave some comments. I'll answer questions for you. I appreciate it. Have a great night.